Hi, everyone. My name's Addie Woicik, and thank you so much to the committee for giving me the opportunity to be here today and talk about our recent work, Gemini, where we use uniqueness-aware network integration of hundreds of gene networks with memory-efficient high-order pooling for improved gene representation learning. So to get started, as we all know, functional screening technologies have become much more accessible in the recent years and really become foundational to our ability to understand biology. And one output of these functional screens can be experimentally derived gene networks, where we get a graph where every vertex represents a gene, and the edges tell us something about the gene-gene relationships that we can discover in that screen. And of course, these gene networks can be really helpful in terms of learning the genetic bases of all sorts of downstream problems we're interested in, including better understanding of human disease and protein function, as well as many other exciting applications we've gotten to hear about today. But of course, one challenge with any of these experimental networks is that almost inherently they're going to be incomplete, and they're going to contain some noisy measurements from that functional screen. So one thing we can try to do to tackle this problem is take advantage of the fact that we don't just have one network. We probably have collections of many, many networks from many different experiments. And we can then perform network integration, where we pool information across all of the networks within a collection to uncover robust and informative patterns about gene relationships. And the problem setting that we specifically try to tackle with Gemini is unsupervised gene network embedding with network integration from very large input collections. So to give you a sense of the scale of input collections that we work with, I'll briefly talk about the two main data sets we use in our work, which are the BioGrid dataset processed into networks by Gene Mania, and the string network collection pre-processed by Mashup. So both of these collections contain sub-collections for yeast, mouse, and human. And in the BioGrid dataset, there are hundreds of networks for each species, with the fewest at 403 in mouse and the most at 895 in human. And string contains 6 networks for each species. Across both collections, the yeast datasets have over 6,000 genes, so 6,000 vertices per graph, and both mouse and human have around 20,000 each. We further assume that all of the networks within our collection cover the same gene set or the same vertex set, but they can be weighted or unweighted, they can be directed or undirected, and it's also perfectly fine, for instance, for some networks to be weighted and others not. Then as output, we produce these dense gene representations that we've learned in an unsupervised way so that they're suitable for many downstream tasks and haven't focused on any particular label during training. So now that I've gone over the problem setting, I want to briefly go over a popular existing method called Mashup that we build off of due to its extreme scalability. And we can fundamentally divide Mashup into 3 main steps. In the first step, given the adjacency matrix representation of each network within the collection, they iteratively smooth this representation using random walk with restart, where after the first step, we have a little bit of information about the immediate neighborhood of each node. Second step, we've sort of expanded that neighborhood size. And of course, we can repeat until convergence, where we get a smoothed understanding of how genes are connected. Then in the second step, once we've produced the smooth representation for every network in the collection, Mashup integrates these by basically taking the concatenated representation. And then finally, once we have this collection-wide representation, Mashup computes the gene embeddings using the truncated singular value decomposition, where we can take the singular values and write singular vectors to combine to form this gene embedding matrix, where in my toy example here, for every gene in the input networks, we've learned a 3-dimensional compact representation. And this is great. It shows us how we can take all of these input networks and get to these dense gene representations. But let's think back about one of our goals in the problem setting, which was to scale to these really large data sets, like the BioGrid human network collection, which is going to have almost 900 networks and almost 20,000 genes. And so if we think about the first step here, storing each adjacency matrix takes on the order of 2.4 megabytes of memory. And once we smooth this and have a dense representation, this is going to be around 1.7 gigabytes. Then in the second step, when we perform this integration, we need 1.4 terabytes to store this concatenated matrix. And then in the third step, because we still need to have access to this collection-wide representation, it's again going to be on the order of 1.4 terabytes. And of course, this can pose a big problem because when we have these really large collections, we can't actually afford this integrated step. And so one of the great contributions of mashup is to employ a mathematical identity so that we can compute the same embeddings, but with much, much lower memory requirements. And the way they do this is by converting this concatenation step that we can't afford to do in memory to a series of summations, which now drops the memory that we need to on the order of 1.7 gigabytes again. And then in the third step, because we've converted this concatenation to a summation, they convert the singular value decomposition into an eigendecomposition, but we can get out the same gene embeddings, now still only needing on the order of that 1.7 gigabytes of memory. And this is great because it makes this network integration tractable for these really large network collections so that we can benefit from all of the data and experimental screens that they contain. But we think there are still some important challenges to think about when we're applying network integration to really large collections. And what we focus on with Gemini is thinking about the sources of variation that exist in those functional screens. So for instance, maybe we think they come from experiments that are measuring different cell types. They might use different assays to quantify gene-gene relationships. Perhaps they're interested in different disease models. And of course, there are many more axes of variation that we might think about existing in these really large collections. And then on top of this, we probably aren't going to have an even representation along all of these different axes. For example, you might imagine that more famous cell lines, perhaps cell lines that are easier to culture, are going to be more represented in these experiments than rarer or more difficult to work with cell lines. And we're probably going to see similar behavior across all of these other axes as well. And so if we think about the sources of variation in these experiments as a really high-dimensional hyperplane, we're not going to have uniform sampling over it. And this means that some networks within our collection may be relatively redundant, or they may be relatively unique. But particularly when there are all of these different sources of variation, we may not have any knowledge about how unique a network is when we're starting the integration. And this brings us to the motivation behind Gemini, where in conventional network integration tools, we often think about these unusual networks as representing noisy and unreliable measurements that we don't want to really impact our learned gene embeddings very much. On the other hand, with Gemini, we think about these unusual networks as representing unique experimental settings that they can actually be particularly valuable because the marginal information add that they can provide to our learned representations might be higher than more redundant networks that we've basically already seen before. And the way we try to take advantage of this is by incorporating a learned network uniqueness term into our integration step so that we can balance the robust patterns that exist in these common repeatable networks with the more novel patterns that we can uncover in the unique networks. As I mentioned before, we build off of mashup so that we can still scale to these really large collections. So in the first step, we also use random walk with restart to compute these smooth representations. Then in the second step, we want to incorporate a notion of network similarity so that we can account for this uniqueness. And so we subdivide this into 3 separate steps. In the first step, something to note is that in order to still perform the similarity computation in a scalable way, we have to perform it between vectors and not between matrices, or it's just going to take too long to be a practically useful tool. And the way we do this is compressing our smooth network representation, which is this very large matrix, into a gene vertex vector, where we learn something about the reachability of each gene node from all other nodes in the graph. And a little bit more specifically, we do this with the 4th order kurtosis pooling of each column in our smooth representation, So intuitively, if we have a gene vertex that is very reachable by most parts of the graph, we might expect that to have a lower kurtosis than a more isolated gene node that is sort of accessible by its clique, but not by most of the graph. And so once we have this vector representation for all of the networks in our input collection, this is now small enough for us to actually compare a lot of these in memory at the same time. And so we can then perform a clustering over all of these kurtosis vectors to identify groups of similar networks as well as the unique networks in our collection. And then finally, in this third substep, we assign each cluster a weight according to its inverse population. So this is basically giving networks that are more redundant lower weights than more unique networks. And then we sample new smooth representations using these uniqueness weights to guide the sampling procedure. So at the end of this step, we get a data set of these smooth representations that is going to more evenly cover that set of variability that we have in the collection. And then finally, once we have this kind of more balanced data set, we can proceed with the third memory efficient step in mashup to perform the summation and then eigen decomposition steps so that we get out these uniqueness aware gene embeddings. So now that we've gone through the motivation and methodology behind Gemini, let's actually look at some results and see if this seems to be helpful. So the way we evaluate the quality of embeddings is with a downstream protein function prediction task using labels from the gene ontology annotation database. I want to emphasize again that this is a downstream task, so we haven't used any of this information when we're actually learning the gene embeddings. For this experiment, we use the biogrid networks produced by Gene Mania. So as a reminder, this is going to have hundreds of networks for every species. And to benchmark Gemini, we compare with mashup, which is the method I talked about earlier, as well as Bionic, which is a recent graph convolutional network. And then finally, we look at the PCA, SVD, and mashup embeddings if we run just over the average of all of the adjacency matrices in the input collection. So first, if we look at the maximum F1 score of that downstream predictor, so higher is better here, Gemini, which is the dark red bar on the far right of all of these plots, is outperforming the other methods over fivefold cross-validation in terms of the biological process, molecular function, and cellular component subontologies. We also see the same behavior if we look at the macro AUPRC of that predictor, so balancing all of the classes equally, as well as the micro AUPRC, where we balance all of the genes equally. And so our takeaway from this experiment was that the uniqueness-aware embeddings are improving this downstream protein function predictor's performance, and they're able to scale to these really large collections like BioGrid. The next question we asked ourselves was whether we could improve the quality of these learned gene embeddings by adding more input networks. And the way we sought to test this was by combining the string and BioGrid network collections. Sort of our thought of what would be ideal here is if the performance of the combined string and BioGrid integration was better than the performance of either collection alone, sort of with the intuition that we haven't removed any information, we've just added additional information. And so now we have this third row where we have this combined network. We take the union of the gene sets from both string and BioGrid, and we're still going to have hundreds of networks because the number of networks is dominated by BioGrid here. So for this, we compare Bionic, Mashup, and Gemini. And first, if we look at their performance run only on string or only on BioGrid, we can see that the methods are sometimes fairly comparable. Bionic and Mashup in particular do well with the string input collection. But really importantly, if we then look at the combination of string and BioGrid, we see that both Bionic and Mashup's embedding quality deteriorates over just using string. On the other hand, if we look at string and BioGrid run for Gemini, we see that the performance continues to improve relative to either collection alone, and that in general, it's the best performing method as well across any input collection. And our takeaway here was that by accounting for the redundancy that exists and sort of the uneven distribution of information when we're combining these two very differently sized data sets, Gemini's embeddings can benefit from this combination. So to wrap up, we really want Gemini to be a scalable tool for the community so that if anyone wants to do downstream work and wants to represent genes, that's a viable option. And so with that in mind, we've released our trained off-the-shelf gene embeddings for the combination of the BioGrid and string collections. And they're currently available on our GitHub, which is linked here as well as in our paper. In the future, we'd like to incorporate some outlier detection tools into Gemini so that we can detect extremely noisy or even potentially adversarial networks. As I mentioned in the talk, we think generally about uniqueness as a good thing that we can take advantage of, and that may be true in some cases, but perhaps it would be useful to say that although the brown network in the center is in its own cluster, but still somewhat near other things, perhaps we should really remove this other network before we proceed with the integration. And then finally, we're also working on an easy-to-use command line tool for the community so that people can apply Gemini's uniqueness-aware gene embeddings to whatever network data set that you're interested in and still benefit from those embeddings. We're also hoping to do this with configurable memory requirements so that you can tell Gemini how much memory is available to you, and we can optimize the computation accordingly. With that, I want to thank my collaborators at the University of Washington who also worked on Gemini. Minjing Zhang was the co-first author of this work, as well as Hanwen Zhu, Professor Sarah Mostafavi, and Professor Shen Wang. And thank you so much for your attention. I'd be happy to take any questions.
Thank you for the great talk. Touching on the very last point with the outlier detection, have you also considered to not take the actual networks from each cluster, but to create like some kind of average network and basically use this to remove noise to some extent? Or is this a bad idea for whatever reason?
No, I think great question. Thank you so much. One thing that I very much glossed over due to time constraints is that when we are performing this sampling, we aren't actually just sampling the original networks or the smooth representations of the original networks. We actually sample the networks in pairs and then use mix-up technique. So then we're basically linearly combining those representations. So I think that helps us a little bit with respect to noise. I think there are definitely additional kind of components that we could do with this that we haven't explored yet, but great question.
Hi, Lars Juliansen. I'm one of the developers of String. So I have to ask, Did you leave out the experiments channel from String when you did this? Because it contains BioGrid, so otherwise things become a little bit funny.
Is this the experimental network? So we kept that one in for the string only integration. So when we're looking just at that, but not for the integrated collections, if that answers.
That makes sense then. And which version of String was it? So I'm guessing effectively you had a newer version of BioGrid than what would be in String itself, which might explain part of the difference.
Yes, I think that is true. Yes.
Other questions? Yes.
Nice. Cool. Thanks for the great talk. So Gemini outperforms its competitors and does so in a really memory efficient way. What's the catch? Are we just getting a free lunch, or are there some limitations? Does it take longer to run? Are there things that you want it to do that it can't do? Or any other reasons to still use competitors or competing approaches?
Yeah, so I think a good chunk of this depends on your data set and sort of some of the assumptions that you have with respect to your data set. So for instance, I think one thing that we see when we're looking just at the string networks, for instance, they tend to be more carefully curated than sort of the BioGrid network that we're using. So if you already have a more even distribution of information, then perhaps this uniqueness aware idea isn't very helpful because it's just already present in your data. So I think we see that with, for instance, a very strong mashup performance there. I also think if you have smaller networks or fewer networks, then methods like Bionic also become much more competitive. For some of the data sets here, you might have noticed that we weren't able to actually show Bionic performance at all because it ran out of GPU memory for us. But if that's an option, then that might be more competitive for you. I think one other aspect is, particularly without this outlier detection component, if you have particularly low confidence, let's say, in your data set, and you really don't think you want to exploit these unique networks, that would definitely be a situation where you should not use Gemini.